Hi everyone, here is the recording of our presentation regarding Pfizer BioNTech COVID-19 vaccine. The name of the student talking will be written on the top of each slide.
In this presentation, we are going to talk about these contents. The ongoing COVID-19 pandemic is caused by severe acute respiratory syndrome coronavirus 2, which has already killed over 2.8 million people around the world. Currently, no effective treatment against this virus is available, although there was a global scientific effort for discovering a potential break of the pandemic, a vaccine against SARS-CoV-2. One of the most promising targets for vaccines and treatments of coronavirus is the spike protein, which plays an important role in viral attachment, fusion, and entry. This enables the virus to insert its genetic material into the host cells and replicate, leading to infection.
Pfizer vaccine, an introduction to its mechanism. The coronavirus vaccine from Pfizer and BioNTech is known as BNT162b2 vaccine, and it is a mRNA vaccine type. The mRNA is made from a DNA template of the virus in the laboratory. The mRNA codes for the RBD, or the building of the S protein. The S protein is used by the virus to attach and infect human cells. It is inserted in the body inside of oily bubbles made of lipid nanoparticles to preserve the mRNA degradation. With the same goal, this vaccine is transported at really low temperatures, minus 70 degrees Celsius. Pfizer vaccine, mechanism and innate immune response. The nanoparticles serve as an adjuvant to attract immune cells to the site of injection by triggering the innate sensors to produce type 1 interferon that is constituted by the interferon alpha and interferon beta, recognized by the IFNAR to stimulate ISGs. Multiple pro-inflammatory cytokines and chemokines are also produced. When the vaccine is injected into muscle cells, myocytes, the phospholipid membrane of the nanoparticle fuses with our cell membrane muscle cells, releasing the mRNA that is translated, originating in the spike protein. Note that the mRNA that enters into the cell cannot enter the nucleus, never having the ability to change our own DNA. The mRNA from the vaccine activates RNA sensors or intracellular pattern recognition receptors, such as TLR7 or MDA5, involved in the recognition of the single-stranded RNA, a pathogen-associated molecular pattern or PAMP, by the innate immune system. Eventually, TLR8, TLR3, and node-like receptors are also activated. Pfizer vaccine, mechanism and adaptive immune response. After mRNA translation and protein synthesis, some fragments of the S protein are presented in the muscle cell surface and can be recognized by APCs, namely DCs, that ingest the proteins, taking them to the regional lymph nodes where they present via the Golgi apparatus the S protein fragments outside the cell in the membrane to a naive CD4+, a cell activating the T cell to an effector T cell that recruits other immune cells. These presentations occurs via MHC2 and CD4 TCR molecules via MHC class II pathway and costimulatory molecules such as CD80, CD86 from the DC link to the CD28 in the T cell, leading to the release of interleukins, which stimulates the creation of memory T helper cells. On the other hand, T follicular helper cells activate B cells when they are presented with the spike proteins. B cells differentiate into memory cells and plasma cells. When the latter secrete antibodies that target the spike protein of the virus, these antibodies, once they get in contact with the virus, mark it for destruction, preventing infection. We've seen the activation of TCD4+ cells through MHC2 pathway. However, APCs can also activate CCD8+ cells to cytotoxic T cells by presenting them a spike protein fragment through MHC class I pathway, where the spike protein was digested by the proteasome. This activation ensures the infected cell's death by the activated killer T cells. About antibody-mediated immunity, according to Dr. Steve Schulman from Israel, this vaccine results in the creation of high-affinity antibodies that neutralize the virus itself, namely IgG and a great portion of IgA, normally produced in infected mucosal tissues, which is good because they provide protection in those places of entrance of the virus like the nose. This vaccine also leads to the generation of immune memory, T and B memory cells. About T-cell-mediated immunity, studies have shown that when a Pfizer vaccine is administered, it leads to the creation in a large scale of cytotoxic or killer T cells that destroy cells infected with SARS-CoV-2. Reviewing mechanism and immune response. From a DNA template, we created lipid nanoparticles with the mRNA from SARS-CoV-2 that codes for the building of the spike protein. The soluble lipid nanoparticles fuse with the lipid membrane of the muscle cells, leading to the translation and spike protein synthesis. The mRNA activates pump receptors such as TLRs. Spike proteins go into the membrane surface and they are recognized by APCs. Innate immune cells are recruited by the release of type 1 interferon due to the NPL adjuvants. APCs in the muscle cells ingest and go into the lymph nodes where they present via MHC2 pathway the antigens to TCD4+ cells activated to T helper cells. Some specific T helper cells, namely follicular helper cells, activate B cells to secrete antibodies of high affinity such as IgA and IgG after somatic recombination on class switching. Through MHC1 pathway, TCD8+ cells are activated into cytotoxic articular cells that are able to destroy infected cells with the virus. Antibodies created from vaccination in the future can easily recognize the S proteins from the virus and trigger a quicker and better immune response.
The development of a safe vaccine normally takes years of research before the product can be deployed to clinical use. However, in present pandemic, scientists worldwide were in a race to produce safe and effective vaccines in record time. So, as we can see here, besides the shortened time in clinical trials and in approval, the long-scale production also started before the authorization phase ends, proving the emergency of releasing the vaccine to the market. Here we can see how vaccines develop and the steps taken to approve them. We are going to focus now on clinical trials, and we have here the clinical trials that took place on Pfizer in order to approve their COVID-19 vaccine. Typically, clinical trials involve three main stages. The fourth stage is continuing monitorization. Phase 1 involves testing on cells and animals and then testing on a small number of people to confirm immune system stimulation. Phase 2 involves testing in hundreds of people, including children and the elderly, further confirming safety in a different group of people, while Phase 3 involves testing in thousands of people. In this phase, scientists see how many people become infected in the placebo and vaccine group. These trials typically determine whether the vaccine protects against the virus with or without side effects and are large enough to reveal the efficiency rate. Based on Phase 3, regulatory authorities decide to authorize the vaccine initially for a limited approval. Regulators review the complete trial results and plans for manufacturing the vaccine and decide whether to give it full approval. As we can see here, the vaccine produced by Pfizer confirmed an overall of 95% effectiveness preventing COVID-19. As we can see here, the immune responses were evident after the first dose and substantially boosted after the second dose. In first phase, the vaccine induced robust SARS-CoV-2 neutralization and S1 binding IgG antibody levels, but immune responses were generally stronger in young group. Also, SARS-CoV-2 neutralizing titers, GMTs, were higher than those observed in people recovered from COVID-19. In phase 2, the vaccine induced robust SARS-CoV-2 immune responses at 1 month after dose 2, defined by SARS-CoV-2 50% neutralizing titers. The COVID-19 vaccines cause a lot of mild adverse effects, as we can see here, after the first and second doses, such as fever, nausea, and muscle pain. The most common is pain at the injection site, and in terms of comparison, the occurrence of adverse effects is lower in Pfizer vaccine than it's in Moderna, as we can see here. Rarely, the vaccine can cause these two types of problem. Anaphylactic shock is a severe allergic reaction. In first contact, the body will produce this type of antibodies. So in the second contact with the same antigen, its connection will release histamine and other mediators that will increase the permeability and distension of blood capillaries. So these are the main consequences generated. Lymphadenopathy is a disease of lymph nodes in which they are abnormal in size and consistency, and were reported and are plausibly related to the vaccine occurred in the arm and neck region. At the end of 2020, the State of Israel had administered more COVID-19 vaccine doses than all countries aside from China, US, and UK. Israel begins to inoculate its population in the end of December last year, as we can see here, and in just over a month it managed to vaccinate about a third of its 9 million inhabitants. More than 82% of citizens aged 60 or over had already been inoculated, and healthy authorities have already started to vaccinate people over 35 in addition to young people. By the end of May, the Israel government expects to have 80% of its population vaccinated. There are a lot of factors that contribute to Israel rapidly and successfully vaccination that could be included in three major groups. related or not related with healthcare system and efforts, as we can see here. Related with healthcare, we have small size, young population, etc. Also, the sophisticated health system, as well as their good health infrastructure and the capability to store and distribute the vaccine, made Israel an attractive country for Pfizer, since the extreme cold storage requirements of Pfizer vaccine were a major hurdle to rapid distribution in many other countries. To join these factors, it was known that Pfizer and Israel government had an agreement, an exchange for continuous and rapid supply of vaccines with data on its patients, although that Israel government guarantees to only give access to aggregated and anonymous demographic data. In addition to that, it seems that Israel paid a higher price for vaccines compared to the European Union. One study that compared people who were newly vaccinated in Israel by Pfizer vaccine with unvaccinated controls showed that the 2 doses of vaccine reduced especially symptomatic cases by 94%.
With the apparition of COVID in its alarming rate of infections, scientists were forced to develop the cure in a very short period of time. For this purpose, many animals were used in order to find the most compatible and closest to the human strain of virus. In Pfizer vaccines, in preclinical studies, BNT162b2 demonstrated protective antiviral effects in rhesus macaques, with concomitant height neutralizing antibody titers and a Th1 biased cellular response in rhesus macaque and mice. For the development of vaccines, the main obstacle to infection of mouse cells with SARS-CoV-2 was the lack of appropriate receptors to initiate the viral infections. SARS-CoV-2 uses the cellular surface protein ACE2 to bind and enter the cells. The mouse ACE2 does not effectively bind the viral spike protein. One strategy to achieve this modification of spike protein is the sequential passaging of SARS-CoV-2 in mouse lung tissue. Another approach to infect mice with SARS-CoV-2 consisted in modifying the mice to express human ACE2. All of these models were very useful for evaluation of vaccines and antiviral agents, and some share features with human diseases. Non-human primate models have been explored for COVID-19 vaccination in rhesus macaques. Studies from several laboratories have shown high level of viral application in both the upper and lower respiratory tract, pathological features of viral pneumonia, and the variable induction of mild clinical disease. There were found evidence of shedding of live virus in both respiratory and gastrointestinal tracts. In addition, hematological changes with evidence of T-cell activation, mild lymphopenia, and neutrophilia have been observed. Immunization of this species with BNT162b2 vaccine resulted in strong antiviral effects against the infection of SARS-CoV-2 challenge. Each animal model has its own merits and demerits, and careful consideration is required before the selection of animal models. The study on transgenic mouse, for example, can provide a proof of concept for understanding pathogenesis. Considering that SARS-CoV-2 infection in non-human primates, like rhesus macaque, most closely resembles that observed in humans, turned out to be a valuable model to evaluate vaccines and drug efficacy. RNA vaccines are progressing into clinical testing for a number of prophylactic and therapeutic applications. This testing allows assessment of their ability to induce robust T cells and antibody responses. The versatile functionality of RNA molecules provide a wide opportunity for future improvements, helping to elucidate the specific RNA elements that have adjuvant activity, thereby informing research towards more potent vaccines. Chemical modification and improved synthesis technologies generate more stable and uniform RNA vaccines. Co-expression of immune-modulating factors and manipulation of the fidelity of RNA polymerase are among the many concepts that may be applied to new generation of RNA vaccines. Another important area of research will be formulation and delivery techniques, which have already been intensely studied in the context of DNA vaccines, allow it to witness the emergence of an entirely new field for effective prophylactic and therapeutic application on large RNA molecules. Even though there is already several vaccines, the pharmacological treatment is also being explored. The figure illustrates the mechanism of action of different drugs against COVID-19. The probable site of drug effect during SARS-CoV-2 infections is one in binding the viral entry via membrane fusion, or endocytosis, which are locked by hydrochloroquine, and 7, the encapsulation and RNA-dependent RNA polymerase, which is inhibited by remdesivir and favipiravir. Studies shown that remdesivir treatment is actually successful against COVID infection. However, the hydrochloroquine treatment is still in studies, and it's not completely sure whether the treatment is successful or not.
Both vaccines contain a part of virus genetic code that is transported into our cells, and they start to produce a spike glycoprotein, which activates an immune response. Pfizer vaccine contains a synthetic version of SARS-CoV-2 mRNA, whereas the AstraZeneca vaccine is a viral vector vaccine which contains a harmless virus altered to contain this code. Pfizer vaccine is most effective than AstraZeneca vaccine against SARS-CoV-2. Another point of view is related to appearance of the new variants of SARS-CoV-2 because of the virus mutations. Until now, Pfizer vaccine may protect against the variant that was first detected in Great Britain, but less effective against the variant first detected in South Africa. The same goes for AstraZeneca vaccine. Both vaccines are administered in 2 doses. AstraZeneca's second dose can be administered 4 to 12 weeks from the first one, and Pfizer is 28 days apart. Regarding the storage of vaccines, Pfizer requires ultra-cold temperature to be stored for 6 months, and in 5 days could be storage in normal conditions of refrigeration. AstraZeneca vaccine is cheaper to make per dose, and it can be stored, transported, and handled in normal conditions of refrigeration for at least 6 months. It's recommended for Pfizer 16 or older, and for AstraZeneca 18 or older. As we can see, Pfizer has higher costs than AstraZeneca. But the question remains, what about side effects? As it was already spoken, Pfizer vaccine has these common side effects, which are similar to side effects of AstraZeneca vaccine, generally resolve within a day or 2 of rest and hydration. Also, Pfizer has a rare but severe reaction that should be monitored after vaccination, but it's treatable, as we've seen before. Although a rare occurrence of blood clotting has been reported after vaccination with AstraZeneca vaccine, and is very similar to heparin-induced thrombocytopenia, so it's called vaccine-induced immune thrombocytopenia. Studies found that this phenomenon was associated with IgG antibodies that recognize PF4 and activate platelets against their Fc receptors, which can be related with hypersensitivity too. Although it's a rare side effect, people who get this vaccine should be aware of the symptoms of this blood clotting problem. Severe headache, visual changes, abdominal pain, nausea and vomiting, backache, shortness of breath, leg pain or swelling, easy bruising. Unlike Pfizer and Moderna vaccines, which are mRNA-based, AstraZeneca and Johnson & Johnson are non-replicating adenovirus vector-based DNA vaccines, and they are associated with these blood clots. In conclusion, different vaccines approach presents different opportunities and challenges.
Our presentation. We leave you here our bibliography if you are more curious about this topic, and thank you very much for your attention. We hope you enjoy it. If you have any question, feel free to ask.